Initial cell. This is our initial cell. DNA protein complex exists in long and thin strands of fibers called chromatin. There would be 46 strands of chromatin in the nucleus of an actual initial cell. However, to be more efficient, we decided to represent them with 4 pieces of long and thin Play-Doh. G1 stage. G1 is a major growth period of the cell. As well, cytoplasmic organelles double in number. It is believed that the centrosome, consisting of 2 centrioles, are replicated during this stage. Synthesis and G2 stage. During synthesis, the chromatin inside of the nucleus is replicated. Then, in G2, also considered late interphase, the cell finally grows to its maximum size, as well as synthesize more species structures in preparation for cell division. Early prophase. In early prophase, the nucleolus completely dissolves, and the centrosomes begin to separate due to non-kinetic core microtubules lengthening and pushing against each other. In the latter part of early prophase, the centrosomes have moved further apart from each other at about 12 and 3 o'clock, and the chromatin fibers have finished condensing into short and thick chromosomes consisting of 2 sister chromatids joined together by a centromere. Late prophase or prometaphase. The nuclear envelope begins to break down, and the centrosomes are now at about 12 and 5 o'clock, almost at opposite ends. The kinetic core microtubules invade the nucleus and start to interact with the chromosomes. Metaphase. By metaphase, the nuclear envelope has completely dissolved, and the centrosomes are now at opposite sides of the cell. The kinetic core microtubules from each centriole attach to kinetic cores on the centromere of the chromosomes and move them to line up at the center of the cell. Meanwhile, the non-kinetic core microtubules continue to interact with each other, stabilizing the cell. Anaphase. The centromeres holding sister chromatids break apart as the kinetic core microtubules begin to shorten. Each chromatid, which are separated to opposite ends of the cell making a V-shape, are now called daughter chromosomes. The cell elongates as non-kinetic core microtubules lengthen. Telophase. During telophase, the nuclear envelope begins to form again, and daughter nuclei containing daughter chromosomes are formed at the ends of the cell. The non-kinetic core microtubules continue to lengthen the cell, and the chromatin fiber starts to loosen in each chromosome. By the end of telophase, the chromosomes are less tightly coiled and a cleavage furrow begins to form. Cytokinesis. The cytoplasm, as well as the cytoplasmic organelles, are separating to the poles of the cell. The cleavage furrow begins to pinch the cell in two. By the end of cytokinesis, 2 separate daughter cells are formed, each with its own set of organelles.